A microscope is a laboratory instrument used to examine objects that are too small to be seen by the naked eye. Microscopy is the science investigating small objects and structure using microscope. Microscopic means being invisible to the eye unless aided by a microscope.
Uses of microscope. This device is used for observing microorganisms and their features. In this field, microscopes are used to study bacteria, cells, and many more. These studies helps biologists in their study of living organisms and their cell structure.
There are the steps in using microscope. First, plug in and turn on the base illuminator. Second, raise the nosepiece using the coarse adjustment knob. This provides greater access to the stage when the slides in positions. Third, rotate the nosepiece so that the 10x objective is in operating positions. You should feel a definite position stop for the objective. Turn the nosepiece clockwise and counterclockwise until you are familiar with this position stop. Fourth, open the iris diaphragm approximately halfway. Fifth, place the slides in the stage slides holder securely. Be sure the slide has the specimen side up. Roll center the specimen over the light coming from the condenser. 6. Move the microscope condenser by means of the condenser rack and pin your knob until the top of the condenser is approximately the thickness of a piece of paper beneath the slide. 7. Lower the objective using the coarse control knob until it reaches a stop. Do not force the knob. The stop should be obvious if you are moving the control gently. Again, become familiar with the feel of position stop. 8. View through the eyepieces and throughout disturbing the coarse adjustment settings, slowly rotate the fine adjustment knob in the appropriate directions until the specimen detail is as sharp as possible. Focus. 9. Adjust the interpupillary distance between the two eyepieces by sliding the eyepiece plates in or out.
10. If your eyes are very different and left eyepiece tube is adjustable to compensate for refraction differences of the eyes, the correct procedure is to bring the specimen into sharpest position focus with a fine adjustment knob, using the right eyepiece only, while closing your left eye to focus for the left eye. View the specimen with the left eye only and turn the knob collar until the specimen is in sharp focus. Do not adjust the fine adjustment knob during this procedure.
Parts of microscope and their functions. 1. Eyepiece. It is also referred to as the ocular. This area is used to view objects through the microscope. It is located at the tip of the microscope. 2. Eyepiece holder. It is often known as the eyepiece tube. The eyepiece is mounted directly over the object lens. Some microscopes, like binoculars, have flexible eyepiece tube that may be turned for optimal visualization and to account for differences in distance. 4. Objective lenses. These are the primary lenses employed for specimen visualization. They have a 40x to 100x magnification range. One microscope has 1 to 4 objective lenses, some of which are forward-facing and other are rear-facing. The magnification power of each lens varies. 5. Nose piece. It is referred to as the revolving turret. It retains objective lenses because it is mobile. The objective lenses can rotate according to the lens magnification. 7. Stage. The specimen is displayed here. The specimen slides are kept in place by stage clip. The most popular stage and mechanical stage, which enable control of the slides by allowing them to be moved mechanically on the stage rather than manual.
Number 8. Aperture. A hole in the microscope stage via which transmitted light from the source enters the stage. Number 9. Microscopic illuminator. It is located at the microscope's base, providing its light source. It collects light from an outside sources. using a low voltage of only 100 volts. It is utilized in place of a mirror. Number 10. Condenser. The lenses used to gather and concentrate light from the illuminator into the sample. They are located near the microscope's diaphragm under the stage. They are essential for producing clear, bright image of high magnification of 400x and more. Number 11. Diaphragm. It is also called the iris. It can be found under the microscope stage, and its primary function is to regulate how much light gets to the specimen. The light intensity and size of that beam that reaches the specimen are controlled by this adjustment device. Number 12. Condenser focus knob. It controls the focus of light on the specimen by raising or lowering the condenser. Number 3. AB condenser. It is condenser mainly created for high-quality microscopes. It allows for extremely high magnification of over 400x. Number 14. Direct stop. It controls how far the stages should move to keep the objective lens from going closer to the specimen's light, which could harm a specimen. It guards against the specimen's light rising too high and colliding with the objective lens.